Do you want to nerd out on diabetes a little bit with me? My name's Bowie. I've had type 1 diabetes for over 20 years. I'm healing naturally, and I love reading research. I read it, I interpret it, I simplify it, and then I find a way to apply it into my own life. I have targeted iron overload as a causative agent in the development of diabetes. And today, I read this paper talking about the pro-radical hydrogen peroxide as a stable hydroxyl radical distributor. Lessons from a pancreatic beta cell. Now, hydrogen peroxide is a typical byproduct of any mitochondrial cellular respiration because every cell has to take out the trash at the end of each workday. Hydrogen peroxide, which is a reactive oxygen species, is that typical trash. It is more stable, less reactive, and able to move throughout the cell without attacking everything. But the stability of the hydrogen peroxide, which initially was a good thing because it's able to move around without being reactive, can turn into a bad thing when there are reactive things in the cell that can make that hydrogen peroxide reactive. Think of it like a super spreader. If there are bad reactive things already in the cell, they use hydrogen peroxide's stability against it because it lets it move throughout the cell, reacting with everything along the way. And that reactive component in the cell is iron. Iron and other heavy metals turn hydrogen peroxide into a pro-radical in the weakly protected beta cells. That means that virtually any protein, carbohydrate, lipid, or nucleic acid is a potential target for the toxic action of the highly toxic hydroxyl radical byproduct of the hydrogen peroxide with the metal. Because we already know that iron has been shown to induce extensive oxidative stress affecting the cell stability and the ability to secrete insulin in beta cells.